Chapter 18, it's about blood. Blood is part of the circulatory system together with the heart and the blood vessels. Cardiovascular system will be only referring to the heart and the blood vessels. Hematology will be the branch of medicine that studies the blood, and some of the functions of the circulatory system will be transporting gases like oxygen, CO2, transporting nutrients, wastes, hormones, and also another function will be to protect by the process of inflammation, which helps to limit the spread of infection, destroy microorganisms, cancer cells, helps to neutralize toxins, and initiates clothing to prevent bleeding. Another function will be regulation. For instance, by absorbing or giving off fluid under different conditions, you can maintain a fluid balance. Also, within the blood, we have buffering of acids and bases to stabilize the pH of the extracellular fluid, and through the blood flow, the cutaneous blood flow, we can control the temperature of our body. Adults generally have about 4 to 6 liters of blood, and blood is a liquid connective tissue that it is composed of plasma, which is considered the matrix of the blood, and the plasma has the following characteristics. So it's clear and it has light yellow color. Some of the form elements of the blood are the blood cells and cell fragments. So within the blood we have red blood cells or RBCs or erythrocytes, we have white blood cells or leukocytes, and we have platelets, which are fragments. They are not complete cells. Some other characteristics of the blood is that they make around 8% of our body weight. In females, we have less volume of blood than males. So we have between 4 to 5 liters, while males have between 5 to 6 liters of blood. And 80 to 85 milliliters per kilogram of our body weight corresponds to blood. And the temperature of the blood will be 1 degree Celsius over the body temperature. So we have 38 degrees Celsius or 104 Fahrenheit. pH of the blood is between 7.35 and 7.45, and the blood is more viscous than water, of course. So the viscosity of the whole blood will be between 4.5 and 5.5. And within the blood we have different molecules that provides a property that we call osmolarity, and we have between 280 and 296 millimoles per liter in the blood. And we have sodium chloride dissolved within the blood that makes 0.9% of the salinity of the blood. Now the percentage of red blood cells that it is found in the blood is known as hematocrit, and in females it is between 37 to 48%, while in males makes between 45 to 52%. Hemoglobin is a protein that carries CO2 and oxygen within the red blood cells, and in females we have between 12 to 16 grams of hemoglobin per deciliter, and in males we have between 13 to 18 grams of hemoglobin per deciliter. The mean red blood cell count in females is less than males, so we have between 4.2 to 5.4 million red blood cells per microliter, and in males is between 4.6 to 6.2 million erythrocytes or red blood cells per microliter. Platelets, we have between 130,000 and 360,000 per microliter. The total white blood cell counts or leukocytes is between 5,000 to 10,000 per microliter. This is a micrograph through light microscopy of the red blood cells. So what you can see here, it's these circular cells that has this edge, this red edge. This will be the erythrocytes. The erythrocytes are more numerous than the white blood cells, and part of the white blood cells will be these big cells that you see here, which are called the neutrophils. These neutrophils increases in number when someone has an infection. And all these small purple fragments that you see here, these will be the platelets. This helps in preventing us from bleeding. So let's talk about the specific components and the general properties of the blood. So the form elements, or the cells, will be known as erythrocytes or red blood cells, RBCs. The other form element will be the platelets, which are fragments that are derived from a cell in the bone marrow that we call the megakaryocyte. And leukocytes will be the white blood cells. And we have 5 different types of white blood cells. These 5 types of white blood cells are subdivided into 2 major categories: the granulocytes, which are the neutrophils, eosinophils, and basophils. And these are called granulocytes because these cells have granules with chemicals or enzymes. The second type or category of leukocytes will be the agranulocytes, which are the lymphocytes and monocytes. These are called agranulocytes because they don't have granules. The prefix in anatomy that it has the A letter means without. So agranulocytes will be without granules, cells without granules. So this is the figure that shows you the different types of form elements. So we have here the erythrocytes, the red blood cells. We have here the platelets, the fragments that prevents us from bleeding. And then we have the white blood cells, which will be the granulocytes and the agranulocytes. So if you see here, this one is a neutrophil, and this is another neutrophil. This is a young neutrophil. It has a nucleus, and this nucleus is continuous. This is known as a band type of neutrophil, or a young neutrophil. These red granules that you see here are filled with enzymes that helps to destroy cells. And this is part of the granulocytes, the neutrophils. This is another granulocyte, the eosinophil. And if you see here, it has this nucleus that is bilobed, so it has 2 lobes connected by this tiny fragment of nucleus. And if you see this in red color, this will be the granules of the eosinophils. Now, the third type of granulocyte will be the basophils. And in the basophils, you can hardly see the nucleus because it has many granules that it doesn't let you to look into the nucleus. So these are then the granulocytes: neutrophils, basophils, and eosinophils. Now lymphocytes, they don't have granules, so these are agranulocytes. And monocytes will be the other type of agranulocyte. So lymphocytes and monocytes, they don't have granules. Monocytes will be the largest white blood cells that we have, and they don't have a granule or granules. When you centrifuge a sample of blood, you can obtain what we call the hematocrit, which will be the percentage of red blood cells that are found in this sample of blood that you have centrifuged. So if you spin this sample of blood at high speed, since the erythrocytes will be the heaviest cells that you have within the blood, they will tend to sink in the bottom of this sample that you have spun in the centrifuge. So you will have between 37% to 52% of the total volume of your sample of erythrocytes settling in the bottom of your container where you have spun your sample of blood. The next layer that you will have in this sample will be the white blood cells, and then the platelets. White blood cells make only 1% of the total volume of the sample of blood, and they will create a buffy coat together with the platelets. The last part of this sample of blood that you have spun in the centrifuge will be the plasma. Plasma will be this fluid that it is light yellow in color with clear tone, and will make between 47 to 63% of your sample. And in the plasma, you have these components. You have water, protein, nutrients, electrolytes, nitrogenous wastes, hormones, and gases. So this is the diagram of, or the figure showing you what happens after you have obtained a sample of blood from someone's vein in the arm, and then you put it in the centrifuge, you spin it at high speed, and then after you withdraw this sample from the centrifuge, you will see that in the bottom of your test tube, you have erythrocytes in the bottom that will make this hematocrit. Then you will have this buffy coat, which is made out of the leukocytes and the platelets, and then on the top you will have this liquid, which is the plasma. And here is the percentages for each of these samples. So 45% for erythrocytes, 1% for platelets and leukocytes, and all of these together will make the form elements, and 55% will make the plasma. So what are the characteristics of the plasma? So it's the liquid portion of the blood, and when you don't introduce a chemical that is known as an anticoagulant in the plasma, you have the formation of blood clots within this plasma. And the remaining fluid, when you have clothed your sample of plasma within the test tube, because you haven't introduced any anticoagulant, and then you remove your fluid, your solids from this plasma, you have a remaining liquid that is known as serum. So serum is the same as plasma, except that it's not going to have the clothing factors and a protein that is called fibrinogen. Now within the plasma, we will have proteins. And some of these proteins will be albumins, which is the smallest and most abundant protein. And we will have also globulins or antibodies. Within the globulins, we have different subcategories. We have alpha, beta, and gamma, and they provide immune system function or protection. Now, one of the functions before we move into fibrinogen of albumins is that albumins provide viscosity to the blood and osmolarity. It will influence blood pressure because it will tend to attract liquid or fluids into the blood vessels. The third type of proteins that you have within the plasma will be fibrinogen. Fibrinogen is a precursor of a thread of protein that we know as fibrin. And this fibrin will help to form a blood clot because it will help to trap red blood cells, it will help to trap platelets, it will help to trap calcium and other cells so you can form a blood clot. Now, all the plasma proteins are formed by the liver with the exception of the antibodies or globulins, which are formed by the lymphocytes or plasma cells. Within the plasma, you not only have proteins, but you have nitrogenous compounds, like the free amino acids that you acquire from your proteins that you ingest, or free amino acids that can be freed up after someone has an injury and you have tissue breakdown. Another type of nitrogenous compound that you can have within the plasma will be nitrogenous wastes like urea, which is the end product of the catabolism of proteins. Now, when you have urea in your blood, the urea will be removed from your body by the kidneys because urea is toxic for our cells. So you have to remove that toxic waste using your kidneys. Within the plasma, we also have nutrients like glucose, vitamins, fats, cholesterol, phospholipids, and minerals. These nutrients will vary in levels or amounts depending on what was your diet. Also within the plasma, we will have dissolved gases like oxygen, CO2, and a little bit of nitrogen. Within the plasma, we also have electrolytes like sodium, which makes 90% of the plasma cations or positively charged ions. So for the viscosity of the blood, the blood, it has these form elements and this plasma with all these components that makes this blood to be a little bit denser, or well, a little bit more denser than water. And this viscosity is going to provide some resistance for the flow of blood. And this resistance will be resulting from the cohesion of the different particles that the blood will have. We have between 4.5 to 5.5 times the viscosity of the blood as water. And plasma itself is 2 times as viscous as water, and it is important in the circulatory function. Now, again, within the blood, we have different ions or solid particles that provides this property of the blood that we know as osmolarity. And blood osmolarity will be the total molarity of the different dissolved particles that cannot pass through the vessel wall. So will be all the trapped molecules that we have within the blood. If you have high osmolarity, your blood will absorb water to decrease this osmolarity. And as you are absorbing water within the blood, you will increase blood volume, which will increase blood pressure. When your osmolarity is low, you won't need as much water within your blood, and your blood pressure will drop because you will have decreased blood volume, and fluids will escape from your blood vessels, and you will have edema, which will be accumulation of fluids outside of your cells in the extracellular space. Some of the molecules that contribute to the osmolarity of blood will be sodium, will be chloride, will be bicarbonate ions, and also glucose. Optimum osmolarity will be regulating all of these elements: sodium, chloride, bicarbonate, uh, glucose, along with proteins and red blood cells. One way that I use to compare blood osmolarity is by thinking about when you are drinking a cup of coffee or a cup of tea. The more sugar that you add to your cup of coffee or your cup of tea, you will increase the density of that coffee or that tea. So you will increase the sweetness, and if you have added too much sugar, more than what you usually like, you need to add a little bit more of coffee or tea, depending on what you're drinking, to decrease that osmolarity or that sweetness. So similarly happens with our blood osmolarity. When you have too much sugar or too much glucose in your blood or too much sodium chloride, you will absorb more water. You increase your osmolarity of your blood, and you will absorb too much water within your blood, and you increase blood volume and you increase blood pressure. And the opposite is going to be true. So if you don't have so much glucose, if you don't have so much sodium chloride or sodium within your blood, you won't need as much water within your blood, and then that water will escape from your blood vessels into the extracellular space, and you will decrease your blood pressure. Now let's talk about erythropoiesis or hematopoiesis, or how the blood is produced. So we have a production of 400 billion platelets, and within this big number we will control our bleeding if someone is bleeding. We also produce between 100 and 200 billion red blood cells and 10,000 billion white blood cells every day. So hemopoiesis is the production of blood, and especially is production of the form elements. Hemopoietic tissues produce blood cells. In the embryo, hemopoiesis happens within the yolk sac initially, so you produce your stem cells or the mother cells that are going to produce the different types of blood form elements. Now, these stem cells in the yolk sac, once they are produced, they are going to migrate to the bone marrow in the fetus, to the liver, the spleen, and the thymus, so you can produce red blood cells in these different places within the embryo. Once we're born, the liver stops producing the red blood cells and the other type of cells, but we will use the spleen to use these cells within the spleen to form lymphocytes. The yolk sac disappears, so we don't have any production of blood cells within the yolk sac because we don't have it anymore, but we will still produce these blood cells within the bone marrow and within the thymus after we're born. So the main producer of form elements in our body as an adult will be the red bone marrow, which is found, of course, within the bones. Within the bone marrow, we have PPSC, or pluripotent stem cells. These were formerly called hemocytoblasts or hematopoietic stem cells. And within these pluripotent stem cells, we will have different categories that are responsible for making the different form elements that we have within the blood. Now a colony forming unit will be a specialized stem cell that produces only a single class of form element within the blood. We have two different categories of hemopoiesis. We have what we call myeloid hemopoiesis, which is the formation of blood within the bone marrow, and we have lymphoid hemopoiesis, which is the formation of blood in lymphatic organs that are outside of the bone marrow. This is a figure showing you these erythrocytes. How does it look like? So the erythrocytes are round cells that looks like a disc, and they have biconcave surfaces. So this is an scanning electron micrograph, a great magnification of these red blood cells. So it can show you this concave surface that it has. And if you see a side view of the erythrocytes within this electron microscopy, you can see the edges of the erythrocytes will correspond to this part, and the central part which will correspond to this part. So you have two biconcave surfaces within the red blood cells. In this panel that you see here, the bottom right panel, you can see this section of a capillary, and this erythrocyte is stuck on the top of each other, forming this continuous structure that we call rholeaux, R-O-L-E-A-U-X. It's like if you were stacking dinner plates on top of each other. So this is the first cell that we will be talking about, the erythrocytes. So the erythrocytes have two main functions. It helps to carry oxygen from the lungs into the cell tissues, and also helps to pick up the CO2 that was generated from the cells on the tissues and bring this CO2 to the lungs so that we can exhale the CO2, because CO2 is a toxic gas. Whenever you have insufficient red blood cells, you can have cell death within minutes because oxygen is necessary to keep the cells alive. The shape of the erythrocytes will be a disc, and the diameter from side to side of this disc is 7.5 micrometers, and the thickness on the edge of this disc will be 2 micrometers. Within the cells, the red blood cells, you have basically most of the organelles of all the cells of the body, with the exception of mitochondria and nucleus. So the red blood cells, they don't have mitochondria and they don't have a nucleus. Because they don't have mitochondria, the cells, the erythrocytes, they cannot produce ATP, because this is the place where most of the ATP is produced. So the cells, the erythrocytes, in order to have energy production, they undergo anaerobic fermentation. Now, since the erythrocytes don't have a nucleus and they don't have DNA, they cannot repair once they have damage. So what it means is that they have a short lifespan. so they cannot undergo mitosis. This is figure 18.4 showing you the surface view of the erythrocytes. So you can see the diameter from edge to edge. So it will be 7.5 micrometers, and this is the thickness on the rim or the edge is 2 micrometers in thickness. In the surface of the membrane of the erythrocytes, you have glycoproteins and glycolipids. These glycoproteins and glycolipids will be important to determine the blood type. And within the cytoplasm, you have cytoskeletal proteins that provides this membrane stability and resilience, and these cytoskeletal proteins will be spectrin and actin. This will help to have the erythrocytes stretching and bending as they're moving through the capillaries. One of the major functions of the erythrocytes will be to transport gases. Because the red blood cells don't have a nucleus and they have this biconcave shape, they can increase their surface area to volume ratio, and this will increase the diffusion rate of gases, oxygen and CO2. Now, within the cytoplasm of the red blood cells, we have hemoglobin, the protein that transports these gases, CO2 and oxygen. 33% of the cytoplasm, or 1/3 of the cytoplasm of the erythrocytes, is occupied by hemoglobin, or Hb. So we have around 280 million hemoglobin molecules on one red blood cell. And the other 2/3 of the cytoplasm can still transport oxygen and CO2, and it helps also to metabolize this oxygen and CO2 so you can have optimal delivery of oxygen into the tissues and optimal delivery of CO2 to the lungs so you can get rid of the CO2. Another important part of these red blood cells is that we have this enzyme that it is known as carbonic anhydrase, or CAH. This carbonic anhydrase will be important to combine or condense CO2 with water to form carbonic acid. And this carbonic acid is going to be important to control the pH, or the hydrogen concentration of ions within our plasma. Hemoglobin, the protein that helps to transport oxygen and CO2, is made out of 4 protein chains known as globins, 2 alpha globins, alpha 1 and alpha 2, and 2 beta globins, or beta 1 and beta 2. In the adult hemoglobin, you have those 2 alpha globins and 2 beta globins. In the fetal hemoglobin, you have 2 alpha globins and 2 gamma globins. So it's alpha and gamma globins in the fetal hemoglobin, and in the adult you have 2 alpha and 2 beta. So the only change is gamma for beta in the fetal hemoglobin. And this change is important because actually the gamma change in the fetal hemoglobin helps to attract more strongly oxygen than the adult hemoglobin, than the beta chains in the adult hemoglobin. And if you remember, the fetuses, they are in the womb of the mother and their lungs are filled with fluid, so they cannot be absorbing oxygen because their lungs, they don't have air in it or oxygen. So they have to compete with the mother's blood for oxygen. And they have to have a stronger hemoglobin the fetal hemoglobin to attract more oxygen molecules into this hemoglobin than the mother's adult hemoglobin. Now, the globins will bind CO2, so 5% of the CO2 in the blood is attached to the hemoglobin. And you also have within the hemoglobin molecule 4 heme groups. The heme groups, they are made out of a non-protein moiety or chemical structure that binds oxygen to iron. So iron is found at the center of the heme group. So this is the figure showing you the components of hemoglobin. So here you have in purple color the alpha chains, alpha 1 and alpha 2, and you have in gold color the beta 1 and beta 2 globins, and in the center of these globin molecules that you see here in red color, you have the heme groups. So if you magnify these heme groups and you look at the chemical structure, you will see these rings made out of these chemical components, but in the center of this heme group you have this iron, and this iron is going to combine with oxygen. So about the quantities of erythrocytes and hemoglobin. So the red blood cells count and hemoglobin concentration can indicate how much oxygen carrying capacity you have. So the hematocrit or the packed cell volume, the percentage of the whole blood made out of red blood cells, is in men between 42 to 52% and in females between 37 to 48%. So we'll be basically indicating the amount of red blood cells in percentages. Whenever you have these percentages of hematocrit, the normal percentages, that means that you can carry a specific amount of oxygen. But whenever you have decreased hematocrit, either in males or females, that carrying capacity of oxygen is reduced. Similarly happens with hemoglobin. So the concentration of hemoglobin in males is between 13 to 18 grams and in females between 12 to 16 grams. If someone has a hemorrhage, the hematocrit and the hemoglobin levels are going to decrease. So that means that the person can transport less oxygen. The red blood cell count is in males between 4.6 to 6.2 million red blood cells per microliter, and in females is between 4.2 to 5.4 million red blood cells per microliter. Whenever this number of red blood cells decreases, the person will have less carrying capacity of oxygen. So you can deliver less oxygen to the cells. Why the values are lower in women than males? Well, it's because we have less androgens, less testosterone. Testosterone in males stimulate the red marrow in their bones to make more red blood cells than females. And also, since females in reproductive stage we have menses, we lose erythrocytes. That's why we have less number of erythrocytes in hemoglobin. And hematocrit is inversely proportional to the percentage of body fat. So females, we have more body fat than males regularly, so that's why we have a hematocrit reduced. In males, they have a greater hematocrit because they have less fat in their body. Let's talk about specifically the production of red blood cells, which is known as erythropoiesis. So we produce around 1 million red blood cells per second. And These red blood cells, they live for around 120 days or 4 months, and their development takes between 3 to 5 days. As the red blood cells are being formed, you have a decrease in the cell size of the erythrocytes, and you have an increase in the cell number, increase in the synthesis of hemoglobin, and as you are increasing the synthesis of hemoglobin, the stem cells that originally had the nucleus within the cells, they're going to kick out the nucleus because you are producing huge amounts of hemoglobin. There is no place for the nucleus to stay within the cytoplasm. Now, the first committed cell in erythropoiesis is the erythroblast. And this erythroblast is part of the erythrocyte colony forming unit. This erythroblast will have receptors for a hormone that it is known as erythropoietin or EPO. This erythropoietin is made by the kidneys. So when this erythropoietin is made by the kidneys and it reaches the bone marrow, it will bind to the erythrocyte colony forming unit cell, and this erythrocyte colony forming unit cell, it will undergo mitosis to form this erythroblast or normoblast that is going to start multiplying and synthesizing hemoglobin. As this erythroblast is maturing, it will form more and more hemoglobin, and then it will discard the nucleus, and you will have these reticulocytes or these immature red blood cells that are going to be released into the circulation, and this reticulocyte is going to have a network of organelles, the endoplasmic reticulum, that ultimately is going to be lost to form a mature red blood cell. Now, within the circulating red blood cells, 0.5 to 1.5% will be reticulocytes. So this is how erythropoiesis happens. So you start here with this hematopoietic stem cell, and this hematopoietic stem cell is going to form this erythrocyte colony forming unit. And this erythrocyte colony forming unit, upon the action of the erythropoietin hormone, is going to form an erythroblast, and this erythroblast is going to go into a series of changes like reducing the cell size, making more hemoglobin, until it reaches a huge volume of hemoglobin within the cytoplasm that there is no place for this nucleus that you can see here. So from this hematopoietic stem cell to erythroblast, you see the presence of a nucleus. But once you have made a lot of hemoglobin in the cytoplasm, there is no more space for the nucleus, so you expel the nucleus from this cell, and then you form a reticulocyte that is going to have this endoplasmic reticulum. So this is going to be a young red blood cell, and ultimately when you lose this endoplasmic reticulum, you will have a mature red blood cell or an erythrocyte. The control of erythrocyte homeostasis, it comes from negative feedback control. So when someone has a decreased number of red blood cells, the person will have less carrying capacity of oxygen. So the person will have what we call hypoxemia, or reduced oxygen levels in the blood. This hypoxemia is going to be detected by the kidney cells, and the kidney cells are going to release this erythropoietin. And this erythropoietin is going to reach the stem cells in the bone marrow, and then you start increasing the production of red blood cells, and you will start forming within 3 to 4 days these reticulocytes or immature red blood cells, and then they will be released into the circulation, and then you increase this number of erythrocytes. So what are the stimuli for increasing the production of red blood cells or erythropoiesis? Hypoxemia, or low levels of oxygen within our blood. When you are in high altitude, because in high altitude we have less levels of oxygen. When you increase your physical activity, so when you exercise, your cells needs more oxygen, so you increase the production of red blood cells. And when someone has lack of oxygenation in the lungs, like when someone has emphysema, they have a destruction of the alveoli, so they can pick up less oxygen, so they have hypoxemia, and then they increase erythropoiesis. If someone smokes or vapes, of course that decreases also the levels of oxygen in your blood, that will increase erythropoiesis. And this is the diagram to show you how this negative feedback loop works. So again, when you have low levels of oxygen in your blood or hypoxemia, your kidney cells and even your liver cells will detect this hypoxemia, and then you produce erythropoietin or EPO. And then that goes into the circulatory system, reaches the bone marrow in any of the bones with red bone marrow, and then you start producing this erythropoiesis, and then you increase your red blood cell count, you increase your oxygen levels in your blood, and then you break down this feedback loop because you reach homeostasis. Okay, now whenever you reach 120 days, or whenever you have damage to your red blood cells, you need to replace them, and you need to recycle them at some point. Now you can have this rupture of the red blood cells, which is known as hemolysis, and this hemolysis happens within the tissue of the spleen and the liver. The tissue of the spleen and the liver has net fibers that helps to trap these erythrocytes. When the erythrocytes are old because they have lost their shape, or when the erythrocytes have been damaged. Now within the spleen, we have macrophages, which are large cells that can eat particles of cells or complete cells. These macrophages, what they will do is digest the membrane of the erythrocytes, and it will also separate heme molecules from globin molecules. The globins, the proteins, will be made into amino acids, and the heme group will be removed, and you will have iron removed from the heme, and the heme will form a pigment that it will be known as biliverdin, which will have a green color. This biliverdin is going to be oxidized later on and be converted into bilirubin or yellow color pigment. And then this bilirubin will be used by the kidneys to produce this yellowish color in the urine, and the liver is going to remove excess of bilirubin. It's going to make bile, and this bile is going to be going into the gallbladder, a little pouch that we have under the liver, that is going to concentrate this bile, and this bile is going to be released into the small intestines, so that bacteria within the intestines, in the large intestine, once it passes from the bile from the small intestine to the large intestine, so it can create this pigment, urobilinogen, that is going to give a brown color to the feces. So this diagram shows you how you have this recycling of the red blood cells components. So the erythrocytes circulate for around 120 days or 4 months. And when the erythrocytes have reached this lifespan and they become old, the membrane becomes more rigid, so there is less flexibility of these erythrocytes. So the erythrocytes will be trapped within this network of fibers within the spleen and the liver, and over there the macrophages, especially the ones within the spleen, will be starting to recycle the different parts of uh, the erythrocytes. So again, the globin molecules will be separated from the heme molecules. The globin molecules, since they are proteins, will be made into amino acids, and these amino acids will enter into the pool of amino acids that we have within the blood, and they will form new proteins. The heme group will have iron, and the iron will be taken up from the heme, and it will be stored within the liver, and then later on will be reused to make more red blood cells, or the iron will be lost in menstruation or injury, and the heme without the iron is going to be converted first into this pigment, green pigment, biliverdin, and then this green pigment is going to be converted or oxidized into this yellow pigment, bilirubin, and then the bilirubin is going to be taken up by the liver and is going to be converted into bile. The bile is going to be stored into this little green pouch that you see here, which is the gallbladder, and then the bile is going to be secreted into the small intestine, and from there is going to go into the large intestine, and is going to make this urobilinogen, and it's going to be used to make the color yellow, brown color of the feces, and it's also going to be used by the kidneys to make this yellowish color of the urine. So basically, our body doesn't waste the components of the erythrocytes. It helps when you are recycling these erythrocytes, it helps to make more proteins and also to make uh, more red blood cells with the iron. Now, if you think about when someone has a bruise or a black eye, you have hemolysis under your skin of red blood cells. So within a few days after you have the black eye or the bruise, you see a change in color from black to green. So basically, this hemolysis, this black color in the bruise or the black eye, it will go the recycling of this heme into biliverdin. So you have first green color, and then with the passing of the days, this green color in the bruise or the black eye will be converted into yellow color, and finally it will disappear because you start absorbing all this pigment from the skin. Just to talk about a little bit of erythrocyte disorders, when someone overproduces red blood cells, is known as polycythemia. Poly means many, cytos means cells, and emia is kind of a name for erythrocytes specifically. We have two different types of polycythemia. We have primary and secondary. Primary is known as polycythemia vera, and this is cancer of this erythrocyte colony forming unit. So this erythrocyte colony forming unit is making excess of red blood cells. So in someone who has this type of polycythemia, it can have a red blood cell count as high as 11 million red blood cells per microliter and hematocrit of 80%. Think about the density of this person's blood. It's going to be very viscous, isn't it? Now, for secondary polycythemia, this will happen when someone has dehydration. So if you lose fluid within your plasma, within your blood, you have more viscous blood. Also, when someone has emphysema, when someone is in high altitude or is doing physical conditioning, is having exercise, or even if they inject EPO. Some athletes, what they do is they inject EPO, erythropoietin, so they can have more red blood cells so they can perform better. Of course, this is cheating. They're going to be caught eventually. So, but this is one cause of secondary polycythemia. And in secondary polycythemia, you have lower number of red blood cells when compared to primary polycythemia, but still is high. It's up to 8 million red blood cells per microliter in comparison to the usually like 5 million red blood cells per microliter that someone usually has. So what are the problems with polycythemia? What are the dangers? Well, you will have increased blood volume, increased pressure and viscosity, which can lead to the formation of blood clots, which are going to migrate to other places of the body. This is known as embolism. The person, some of these blood clots can occlude some of these narrow blood vessels, the tiniest blood vessels, which will lead to a stroke, or the person cannot pump as easy the blood with the heart, so it will result in what we call heart failure. It's not the only cause of heart failure, but this can be one of the consequences of polycythemia, developing heart failure. It's like if you this analogy of heart failure is if you have a very thick blood, it's like if you have added into your four-cylinder car the oil. If you change the oil into of your four-cylinder car, if you add oil for a truck or a diesel vehicle, the density of this oil is going to be very viscous, that if you add it into your four-cylinder car, which doesn't require a very viscous oil or very dense oil, you're going to burn your engine. So this is what happens with heart failure. Okay, so another problem that it can happen with the production of red blood cells is that possibly you don't produce as many, and this is known as anemia. So anemia is inadequate erythropoiesis or hemoglobin synthesis. Some causes of anemia will be kidney failure, like what happens in end-stage organ damage in someone who has diabetes. So if you don't have healthy kidneys, you don't produce as many erythropoietin. So your red blood cells won't be produced. Also, since iron is necessary for the formation of hemoglobin, if you don't ingest iron, you don't produce as many red blood cells. So this will be known as iron deficiency anemia. Another problem is the lack of vitamin B12 or ingestion or absorption or attack of stomach tissue of some cells that are going to protect your vitamin B12. So vitamin B12 is necessary for the formation of red blood cells. So when you don't absorb vitamin B12, when you don't ingest vitamin B12, or when you don't produce a protector of vitamin B12, which is known as intrinsic factor, you will have what we call pernicious anemia. Also, when you lack folic acid, when you don't have folic acid in your diet, you will have anemia. Another problem, it will be the decreased production of red blood cells by what we call hypoplastic anemia. In this case, you still produce red blood cells, but you produce it at a slower rate. Or you might not produce red blood cells at all, and this is called aplastic anemia. Why this can happen? Well, if someone has leukemia or cancer of the bone marrow, okay? So you can have reduction of the production of the red blood cells. Another cause of anemia will be an anemia because you have bleeding. And it can be because you have an injury and you can have a sudden bleeding, or it can be a chronic bleeding. What if someone has an ulcer and that ulcer is bleeding constantly? So this will be a chronic bleeding. Another problem is that you can have anemia because you are destroying a lot of red blood cells. When someone has an enlargement of the spleen, because that can happen in certain diseases, you start over destroying red blood cells. Or what if someone has a crash injury and it hits a big area of the body and a lot of red blood cells were destroyed? This is known as hemolytic anemia too. Okay? So what are the consequences of anemia? Well, since you have less red blood cells, you carry less oxygen to the cells, so you have tissue hypoxia, so your cells will be starving for oxygen. And if this happens for a long period of time, your cells will die. Abnormally will be dying. This is known as necrosis or infarct. Some of the symptoms of anemia is that the person will be lethargic or will be less energetic, so the person won't have energy to do things. The person will have dyspnea or decreased oxygenation or shortness of breath upon exertion. And the person can have necrosis or infarcts in the brain, the heart, and the kidneys, or other tissue, but these are the three most important tissues in our body: the brain, the heart, and the kidneys, or organs. Okay. Someone who has anemia, you will have reduced osmolarity, and this will lead to edema. Now, when someone has anemia, the blood will be less dense, so you have reduced viscosity of the blood, and the person will have heart races, decreased blood pressure, and the person will have cardiac failure. Another problem will be sickle cell disease of the production of the red blood cells. So in this case, this is a hereditary defect that happens mostly among people of African descent, and in this case, the person will have a mutation in the gene that codes for the protein hemoglobin. This is caused by a recessive allele. We will see this more than likely towards the end, again towards the end of the semester or the course. So in this case, the person has to have a mutation in both genes for hemoglobin coming from both parents. And the mutation is in a single amino acid. So in the 6th amino acid of the beta chain. Whenever someone has a mutation in this hemoglobin, because it has sickle cell, the hemoglobin doesn't bind well oxygen. And this hemoglobin that it is mutated now is going to be called HbS, or hemoglobin sickle cell. The red blood cells in the person who has sickle cell disease are not going to be as flexible, so they are rigid, and they will have not the circular shape of a disc. It will have an elongated shape that ends in points or sticky pointed ends. Another problem is that these erythrocytes have the tendency to clump together much easily, which makes them more likely to block small blood vessels, and the person will have consequently more chances to having uh, stroke, heart failure, kidney failure, joint pain, or paralysis. Now, in some persons, the, the sickle cell allele happens only in one gene coming from the mom or the dad. This is known as heterozygous. And in this case, the person will be resistant to malaria because it doesn't have the receptors for this malaria parasite. So it will have a protective function. In this figure, they are showing you two different types of erythrocytes. The normal erythrocytes that you can see here with this circular shape, and the sickle cell erythrocytes, which have this shape of a sickle. A sickle is an instrument that they use in agriculture to cut the grains of wheat. So this is the ending of a sickle of an instrument. So it looks like this sickle cell shape looks like a sickle. Okay, so the red blood cells have in their surface, in the plasma membrane, different proteins. And these different proteins helps to classify the erythrocytes or the blood into different types. So blood types and transfusion compatibility are a matter of the interaction between these plasma proteins known as antibodies and the antigens found in the erythrocytes. So blood types are based on interactions between antigens and antibodies. Antibodies are the proteins that we have in the plasma that protect us from different attacks. And the antigens are found in the normal cells, including the erythrocytes. So what are antigens? Antigens are complex molecules that are found in the surface of the cell membranes of cells, including the red blood cells. And these antigens, when they are not recognized as their own, they can activate the immune system, and then you can have attack of the immune system cells to these different cells. Now, the antigens in the blood are genetically inherited, and they are unique to the individual. Now, these antigens are usually used by the immune system to distinguish the self-antigens from foreign antigens or foreign matter. The foreign antigens will generate an immune response, while the self-antigens will not. And agglutinogens will be the specific name of the antigens found in the surface of the red blood cells, and this will be providing the basis for the blood typing. What are antibodies? So the antibodies are proteins or gamma globulins that are secreted by the plasma cells or B lymphocytes. These antibodies are part of the immune response to foreign matter or foreign antigens. So when the antibodies find an antigen, they can bind to this antigen and they can mark them for destruction. And when they do so, when they bind to these antibodies, they will form a complex known as antigen-antibody complex. And the other name for the antibodies are known as agglutinins. These antibodies, these agglutinins, are found in the plasma, and that bring about transfusion mismatch. when you have clumping of antibodies to the antigens, or they're forming these antibody antigen complexes, they will form a heavy complex that will agglutinate and will cause the clumping of the red blood cells. So, red blood cells with their antigens in their surface can form these complexes with these antibodies. And the antigens in the red blood cells are called agglutinogens, while the antibodies are called agglutinins. Now, the antigens are called antigen A and B and also D. These are determined by glycolipids on the red blood cell surface. And the antibodies found in the plasma, the agglutinins, will be anti-A, anti-B, or anti-D. So, this diagram shows you these antigens in the surface of the red blood cells. So in here we have type O red blood cells with these different molecules that makes this antigen. So we have N-acetylgalactosamine in red color, galactose in yellow color, and fucose in blue color. These will be sugars and glycoproteins in the case of N-acetylgalactosamine that will form a complex or an antigen. So type O blood has only this antigen. Type A has a similar antigen to type O, as you can see here, the red, yellow, and blue color, plus it has this N-acetylgalactosamine. Type B is similar again to this type O, but it has this extra galactose. And type A and B, blood type, which is here, it will have this A-type antigen, which you can see here, and it has this B antigen that you can see here. So this is type A, type B, and type AB. So the ABO group classification is determined by the presence of these agglutinogens that we already saw in the previous slide. And a blood type A person has A antigens only, while a blood type B person has B antigens only in the red blood cells. And someone who has AB blood type will have both A and B antigens. And someone who has blood type O, it doesn't have A and it doesn't have B antigen. So the most common type of blood that we have is O type, and the rarest type will be AB. When you are performing just the ABO blood typing, you will use antibodies. In the case of this left side column, you will use anti-A, and in the case of this B-type column, you will use anti-B antibodies. So you will, using a lancet, you will prickle the tip of the finger of someone, and you will be adding into a plastic card, into circles that are found within this plastic card, some drops of blood into this plastic card. And this blood will be combined with this serum or these antibodies that you have. So if you are testing for type A blood, you will add these anti-A antibodies, a little drop, and then you will mix this blood with these antibodies. You will mix it really well and wait for agglutination to happen, or clumping. So if you see here in this part, you have clumping. These little granules or clumps that you see here, that indicates that the person has an A antigen in the surface of the red blood cells, and these A antigens were bound by these anti-A antibodies. If the person is only A type but not B, the person won't have clumping when you have added into this drop of blood the anti-B antibodies. So in here, you don't see any clumping. For B type, you don't have clumping with anti-A antibodies, but you see this clumping with anti-B antibodies. So this person has B type. If the person has AB type of blood, the person will have clumping in A and clumping in B. And if the person is O type of blood, you won't have clumping of O and clumping with with A and clumping with B. So you don't see any clumping. Now, along with this, you have positive and negative groups of ABO types. So someone can be positive or negative for A, positive or negative for B, positive or negative for AB, and positive or negative for O type of blood. Now, this positive or negative, it comes from another antigen that it wasn't shown in the previous slide. This antigen is known as D antigen or Rh antigen. And this also can be tested with antibodies that is not shown in here, but are known as anti-D antibodies. So when someone has positive type of blood or is Rh positive, the person will have another column in here with a drop of blood, and then you mix this drop of blood with these antibodies, and then the person, if it's Rh positive, will have clumping with the D antigen. Or sorry, D antibodies and antigen. Okay? So if someone is negative, you don't see any clumping with the D. And that will be for any type. So if you have type A, positive will be clumping in here. If you have type A negative, no clumping in here, and you keep on going. So within your plasma, depending on the type of blood that you will have a specific type of antibodies or not, or agglutinins. So if you have B type of blood, you will have anti-A antibodies, but you don't have anti-B antibodies because you cannot attack your own blood. If you have O blood type, you will have no antigens in the red blood cells, but you can have anti-A and anti-B antibodies. Now, the agglutinins or the antibodies will appear 2 to 8 months after birth, and the maximum concentration will reach by 10 years of age. So antibodies A or antibody B, or both or neither, are found in plasma depending on what type of blood you have. And you don't form antibodies against your antigens. Again, if you have A type of blood, you cannot have anti-A antibodies. You can have anti-B, but never anti-A, because you cannot attack your own blood. If you have B, blood type, you cannot have anti-B. You can have anti-A, but not anti-B. And if you have AB blood, you cannot have anti-A and anti-B antibodies. So you don't have neither antibodies. And if you have O type of blood, you will have both anti-A and anti-B antibodies. And this diagram is showing you the antibodies, how they can bind erythrocytes. So in this case, this will be pentamers of antibodies, so groups of 5 antibodies that can bind many erythrocytes at once, forming agglutination of the erythrocytes or clumps. So agglutination, each antibody can attach to several foreign antigens on several different red blood cells at the same time. So that's why they are pentamer, because they can grab up to 5 erythrocytes. And since you have many antibodies, you can create a clump or agglutination. And these antibodies are responsible for mismatched transfusion reactions. So the agglutinated red blood cells can block several small blood vessels, and depending on the organ, it's going to be the signs and symptoms that you will have, but most of the organs that will suffer because of a transfusion reaction will be brain, kidneys, and the heart. And these antibodies, once they bind to these antigens, they will set an immunological reaction to destroy these foreign red blood cells or foreign antigens in the red blood cells. So you will have hemolysis, and you will start releasing hemoglobin over the next few hours or days, and then you will have the conversion of this heme group into biliverdin, bilirubin, and then you have this yellowish color in the skin. And more than likely, hemoglobin, when it's free up in the circulatory system, in the blood vessels, it will block a structure in the kidneys that are called the kidney tubules, and then the person will have renal failure, acute renal failure. So their kidneys won't work. So this is to show you what happens with transfusion reaction. In this case, you have a type B recipient. So the person have B type of blood. And through a mismatch, the person received type A blood. And since you have these antibodies are going to start clumping these A-type blood erythrocytes, and then they will start forming this agglutination or these clumps that are going to block these small blood vessels. Why you don't have block of this large blood vessel? Because it's too large to be blocked by these agglutinating complexes. But in here, you have the agglutinated red blood cells blocking these small blood vessels. If you were the kidneys, you will have acute renal failure. So within these different types of blood that you can have, A positive, B positive, O positive, or AB type, negative, positive, etc., you have a couple of subclassifications. You have what we call the universal donor, in which the type O blood, which is the most common type, will be called the universal donor because their red blood cells doesn't have antigens. And the the donor's plasma may have both antibodies against the recipients, anti-A and anti-B, and may give packed cells minimal plasma. But when it comes to the red blood cells, the person doesn't have antigens, so it can donate to any type of blood. So it can donate to A, B, AB, and specifically the universal donor will be negative type, O negative, not O positive. It's O negative, the universal donor, because it won't have the antigen or Rh factor. Now the universal recipient will be AB positive. This is the rarest type of blood, and in this case the person can receive any type of blood because it will have A antigens, it will have B antigens, it will have D antigens, the AB positive. And it doesn't have antibodies because it cannot attack its own blood. So it doesn't have anti-A, anti-B, or anti-D. So the Rh group, this antigen that I already talked about before, what it makes you positive or negative. There is more than the antigen. We have RHC or RHE, but the most common type is RHD. And this Rh group, it takes its name because it was first discovered in 1940 in a type of monkey that is called rhesus monkey. And the RHD, since it's the most common type, it will make the person to be tested for this, the antigen, together with the A, B, or O classification. So again, someone can be A positive or A negative. Someone can be B positive or B negative. Someone can be AB positive or AB negative. And someone can be O positive or O negative. If someone is positive, that means that their erythrocytes have the D antigen on their red blood cells. And if someone is Rh negative, it doesn't have this D antigen. And the Rh frequencies vary among ethnic groups. So, anti-D agglutinins are not normally present. And when they are present, they will be present in persons who are Rh negative type and has been exposed to Rh positive blood. For instance, if a female is Rh negative in their blood and it gets pregnant and the fetus is Rh positive, or this female or even a male has received, if it's an Rh negative type of blood in a male, if this male has received a transfusion with Rh positive type of blood, this person will start developing antibodies against the Rh group. There is no problems within the first transfusion or the first pregnancy, but in subsequent pregnancies, if the fetus keep being Rh positive, or if the person received again a blood transfusion with the Rh positive type, the person will develop a transfusion reaction. Now, in the case of a female who's Rh negative and the fetus is Rh positive, the first fetus that she got pregnant with is Rh positive, she will start developing antibodies. And in subsequent pregnancies, the female will have preformed antibodies if she hasn't received a vaccine that is called ROGAM. So she will have preformed antibodies that are going to go through the placenta and are going to destroy the red blood cells of the subsequent pregnancy of Rh positive fetus. So there is a way then to prevent this from happening. If a female is Rh negative and it gets pregnant for the first time with a baby whose blood is Rh positive, she can receive this ROGAM vaccine, which is an antibody that is going to bind to her antibodies against this anti-D, preventing these anti-D antibodies reaching the fetal blood and destroying the fetal blood. Now, if this doesn't happen, then if this female doesn't receive this ROGAM and she has a second pregnancy with an Rh positive fetus, her antibodies will cross the placenta, will bind the red blood cells of the baby, and it will start destroying these red blood cells of the baby in what we call hemolytic disease of the newborn, or HDN. And of course, the baby will be anemic and the baby can die. So this diagram shows you the hemolytic disease of the newborn. So this is the first pregnancy. See, this female is Rh negative, the baby is Rh positive. During labor and delivery, part of the blood of the baby will cross into the circulatory system of the mother, and she will start developing antibodies against the baby's blood. But since the baby has born, there is no way how this transfer can happen so rapidly that the first baby will have destruction of the red blood cells. It's only in this second pregnancy where she has already formed these anti-D antibodies or anti-Rh antibodies that she will have them ready. And these anti-D antibodies will cross the placenta in the subsequent pregnancy with the Rh positive fetus, and then this baby will have destruction of these red blood cells. So this is hemolytic disease of the newborn. So there is this mismatch of blood type between the mother and the baby. Now, around 15% of the population is Rh negative. So the likelihood for this event to happen is rare, but since there is 15% of the population who's Rh negative, and if the female is Rh negative and she's in reproductive age and she's going to have a baby, imagine what are the possibilities for her to have a couple who's Rh negative, so that the chances are very little. Okay, so that's all about the red blood cells. Now let's move into the white blood cells, or the leukocytes. So the leukocytes are the least abundant form element. So we only have between 5,000 to 10,000 white blood cells per microliter. Why is this? Well, because in general the function of the white blood cells is to protect us against infections of microorganisms or other pathogens or disease-causing organisms, or even cancer. Since we are not always having infections, we don't require as many white blood cells as red blood cells. We are always requiring the transfer of oxygen and CO2. So we need to have more red blood cells than white blood cells. Now, in comparison to the white blood cells, to the red blood cells, the white blood cells do have a nucleus, and that nucleus is large, it's conspicuous. And the white blood cells spend only a few hours in the bloodstream before migrating into connective tissue. And usually that migration or that movement of the white blood cells into the connective tissue happens because someone might have a problem in the connective tissue, like an infection, like an injury. But if not, there is very hardly migration of the leukocytes towards the connective tissue. Now, the white blood cells, they retain their organelles for protein synthesis, and they retain their nucleus. And some of the white blood cells, they have granules. Okay, and all the white blood cells have some lysosomes called non-specific granules, or acidophilic. Now, the granulocytes have specific granules that contains enzymes or other chemicals that helps in the defense against pathogens. So again, which are the granulocytes? Neutrophils, eosinophils, and basophils. And the percentages are there, so 60 to 70% makes the neutrophils, 2 to 4 the eosinophils, and less than 1% the basophils. Characteristic of the neutrophils, their nucleus have a different shape. That's why they are called polymorphonuclear leukocytes. Poly means many, morphos means shape, nuclear means nucleus. And the neutrophils, they have barely visible granules in their cytoplasm, and their nucleus have between 3 to 5 lobes. The eosinophils, they have many granules, and their color of their granules vary in color between rosy to orange color. And they have a nucleus that has 2 lobes. They are bilobed. The basophils, they have these large abundant violet granules, and this color and their abundant granules obscure a large S-shaped nucleus. Now for the granulocyte, the lymphocyte, they make between 25 to 33% of the white blood cells, and they have variable amounts of cytoplasm. And actually, their nucleus is so large that almost occupy the entire cytoplasm. So the lymphocytes are very easy to identify because it's more nucleus than cytoplasm in the microscope. Now the monocytes, which makes between 3 to 8%, are the other type of granulocytes. They are the largest white blood cells, and they have an ovoid, kidney, or horseshoe-shaped nucleus. And the monocytes will be precursors of the big eater cells that we call the macrophages. So in functions, the neutrophils are antibacterial, so they increase in number when someone has a bacterial infection. The eosinophils, they protect us from parasitic infections, and they increase in numbers when someone has diseases of the collagen, which is a protein of the connective tissue, among others. Also, they increase in number when someone has allergies or when someone has diseases of the spleen or the central nervous system. Functions of the eosinophils would be to phagocytose antigen antibody complexes, also to phagocytose allergens, and they release inflammatory chemicals. And also they release enzymes to destroy large parasites. Basophils, they increase in number when someone has viral infections like chickenpox, when someone has sinusitis, when someone has diabetes, and they secrete chemicals like histamine, which is a vasodilator, so basically enlarges the diameter of our blood vessels and speeds the flow of blood to an injured area. Also, they secrete an anticoagulant that is called heparin that promotes the mobility of other white blood cells in the area. So this is how the granulocyte looks like. So this will be the neutrophils. As you can see, they have 3 to 4 nucleus in this case, and that's why they have different shape. That's why they're called polymorphonuclear. Now the eosinophils, they have this bilobed nucleus, and they have this rosy orange. They have these rosy orange granules. Basophils, they have these pink granules. They are very abundant, and as you can see, the nucleus is barely seen because of that. So lymphocytes, they increase in number when someone has many diverse infections or immune responses. Functions of the lymphocytes in general will be to destroy cells like cancer cells, foreign cells, and virally infected cells. And another function is to present antigens to activate the other immune cells and also help to coordinate the action of other immune cells and the B lymphocytes specifically, which we will see later on in the immune system chapter. But these lymphocytes, the B lymphocytes, are known as plasma cells, secrete antibodies, and they help to provide immune memory. Monocytes, they increase numbers in viral infection and chronic inflammation, and they leave the bloodstream and transform into macrophages. So the monocytes are precursor of this big eater of macrophages, and these big eater macrophages phagocytose microorganisms or pathogens and cellular debris. They help also to present antigens to activate other immune cells as part of the cells that we call APCs or antigen presenting cells. All of these terms we will see it in again, and you understand it better in the immune system chapter. So this is how this agranulocyte looks like. So this is the monocyte. This is the largest white blood cell. It has kind of this horseshoe shape or kidney shape and nucleus. And this will be the lymphocytes. Remember what I told you, the lymphocytes have a very large nucleus and very little cytoplasm. Production of white blood cells is known as leukopoiesis, and the production of the white blood cells start with the hematopoietic stem cells or HSCs. So these HSCs will transform or differentiate into myeloblasts to form neutrophils, eosinophils, and basophils. So myeloblasts will form the granulocytes. And when these HCS or hematopoietic stem cells differentiate into monoblasts, it will form monocytes. And when these HCS differentiate into lymphoblasts, it will form the lymphocytes. So we have two types of lymphocytes. We have B lymphocytes and we have T lymphocytes. The B lymphocytes and the T lymphocytes form within the bone marrow, but the B lymphocytes mature within the bone marrow and the T lymphocytes mature within the thymus. Now, the red bone marrow will store and release granulocytes and monocytes. The circulating white blood cells do not stay in the bloodstream. The granulocytes will live in 8 hours and live 5 days longer. Monocytes will live in 20 hours and will transform into macrophages that can live for several years. The lymphocytes, they are in charge of providing long-term immunity, so they live for as long as you live. So they are finite in number, so that means that the ones that you form and that you will be living with will be the only ones that you have. So if your lymphocytes get destroyed for some reason, like if you have an infection, they will not get back. And these lymphocytes are continuously recycled from the blood to the tissue fluid to the lymph back into the blood. So they circulate between the lymphatic system and the circulatory system. So this is the diagram showing you the different lineages of the white blood cells, how they form. So you have the hematopoietic stem cell that is going to differentiate or specialize into the different colony forming units, including the eosinophilic colony forming unit, which is going to form the eosinophils, the basophilic colony forming unit, which is going to make the basophils, the neutrophils, the monocytes, the lymphocytes. Okay, so you have B lymphocytes, T lymphocytes, and NK cells, and all of these monocytes, neutrophils, basophils, and eosinophils, which will form all together the white blood cells or leukocytes. Some disorders of the leukocytes. So someone can have low count of leukocytes, which is known as leukopenia, and in this case, the person will have less than 5,000 leukocytes per microliter. Causes for leukopenia will be if someone were subject to radiation, so that will damage the stem cells in the bone marrow, and consequently the person won't form as many white blood cells. Certain poisons also damage the bone marrow. Infectious diseases can destroy white blood cells, like in the case of of HIV. It can destroy the white blood cells. So what are the consequences of having leukopenia? Well, increased risk of infections. The opposite of leukopenia will be leukocytosis. In this case, we'll be having a higher count of white blood cells above 10,000 white blood cells per microliter. Causes will be having infection, allergies, certain diseases. And in order to know specifically what white blood cell is increased, you have to do what we call the differential white blood cell count, that will identify which of the leukocytes is increased in percentage. And that will pinpoint to what is the specific problem. Another problem of the leukocytes will be leukemia. In leukemia, your stem cells in the bone marrow start overproducing leukocytes. So that will increase the number of circulating leukocytes. And this increasing number of leukocytes is going to be way higher than leukocytosis. So someone can have, let's say, 100,000 white blood cells per microliter. Does this makes the person super immune? No, it isn't, because actually the white blood cells that are produced by these abnormal hematopoietic stem cells are going to be defective, so they are going to be non-functional. It's like if you are, or someone is a millionaire, but it has fake money. This person might try to go and purchase something with a lot of money, but if they discover that this money is fake, of course the person is going to go to jail. So it's dysfunctional or non-functional money. So similarly with leukemia, the person will have huge number of leukocytes in their bloodstream, but those huge numbers are going to be non-functional. So we can have different types of leukemia depending on the white blood cell that it is overproduced. So we can have myeloid leukemia, in which you have increased number of granulocytes, which will be basophils, eosinophils, or neutrophils. You can have increased number of lymphocytes. In this case, you have lympholeukemia, and you can have increased number of monocytes too. And you can have acute leukemia or chronic leukemia. In acute leukemia, you have basically sudden increased number of leukocytes, and this leukemia progresses rapidly and the person will die within months. In chronic leukemia, it's all the opposite, so it's slow in progression and the person can have survival time. I mean, your book mentions, and even here in the PowerPoint, it mentions 3 years, but it's up to a certain point not valid, because you can never tell specifically what is the survival time. Okay, now the effects is that the normal cell percentage of every single type of white blood cell or red blood cell is going to be disrupted. Why? Because even for the platelets. Why? Because this abnormally growth of stem cells within the bone marrow is going to prevent the normal cells from doing their function. So you have decreased number of all type of blood cells, including the platelets. And this is how the normal blood smear will look like, and how an abnormal uh, blood smear will look like. So all of these will be leukemic cells. So there are many, and they are abnormal. So the complete blood count, or CBC, includes several values. It will include hematocrit, hemoglobin concentration, your total number of red blood cells, reticulocytes, white blood cells, including the differential, the platelet numbers, and it will include also the size of the red blood cells and hemoglobin concentration per red blood cell, so you can have a good diagnostic tool. Now let's talk about, we're done with the platelets, sorry, with the white blood cells. Now let's move into the platelets. So the platelets are fragments of cells that are derived from the cytoplasm of the megakaryocyte. Megakaryocyte is a big cell that is found within the bone marrow and never leaves the bone marrow, only sheds these pieces of cytoplasm that we know as platelets. And the platelets are important in hemostasis, which is the cessation of bleeding. So these are important then to stop potentially fatal hemorrhages. And we have three hemostatic mechanisms. So when someone has an injury into the blood vessels are going to constrict, or they are going to reduce their diameter. This is known as vascular spasm. And then afterwards, you will start forming a platelet plug, and then you will start forming a blood clot through the coagulation cascade. And platelets play an important role in every step of the hemostatic mechanism. Again, the platelets are small fragments of the megakaryocyte cells, and their diameter is between 2 to 4 micrometers, and they have granules. Within these granules, they have chemicals that helps to prevent overbleeding. And the platelets also have a canalicular system. So it's a complex of channels inside the platelets that helps to deliver these chemicals that they have in the granules into the outer surface, so that they can start forming these platelet plugs. And the platelets, once they start functioning, they have this amoeboid movement and phagocytosis. Normal counts of platelets is between 130,000 to 400,000 platelets per microliter. So functions. They secrete vasoconstrictors, so they can create this spasm in the blood vessels to help to reduce the diameter of the blood vessels, so you can have less hemorrhage or leaking of blood. They will stick together to form this platelet plug that will form a temporary seal within small injuries within the blood vessels, and they will secrete chemicals that act as procoagulants, or clotting factors that promotes clotting. And then they will initiate the formation of a clotting dissolving enzyme. And they will attract neutrophils, monocytes to the site of inflammation, and then they phagocytose and destroy bacteria, and will secrete growth factors that will stimulate the repairing of the blood vessels and mitosis of the cells of the blood vessels. So this is a microscopy to show you the granules inside the platelet. So all of these are granules, and these extensions of their cytoplasm are known as, of the membrane in the cytoplasm are known as pseudopods. And they also have mitochondria and the open canalicular system. So this is a cartoon showing you how the platelets are shed into the circulatory system. So this is the megakaryocyte in the bone marrow, and this is a capillary in the bone marrow that is known as sinusoid, and the megakaryocyte extends within this sinusoid, this cytoplasm extension, and then it starts fragmenting, forming proplatelets, and then these platelets, and then you have platelets within the circulatory system. Formation of platelets is known as thrombopoiesis, and the stem cell that makes these platelets is known as megakaryocytes, and they act upon the action of a hormone that is called thrombopoietin. So the megakaryoblast, they will, under the influence of thrombopoietin, will undergo replicative DNA without dividing, and then they form this gigantic cell known as megakaryocyte, and this megakaryocyte will live in the bone marrow adjacent to the sinusoids, and then they will extend these fragments or proplatelets that protrude between the sinusoid cells, and then they will split off fragments called the platelets. And the platelets will circulate freely between 5 to 6 days in the circulatory system, and 40% will be stored in the spleen. And this is the mechanism for hemostasis. So if you have a cut in one of your blood vessels, these fibers from this cut will get exposed into the inner diameter of the blood vessel, which is this, and it will start activating these platelets, and these platelets will secrete these chemicals together with chemicals that are released by the injured cells within the blood vessels, and then you have a spasm of the blood vessel, and you have vasoconstriction. So you have reduced the diameter of this blood vessel, so you can prevent bleeding. Later on, these platelets will form a platelet plug, which is going to be a temporary seal, and then this will activate a cascade, which is going to make these proteins that we have within these blood vessels that are called fibrinogen to form fibrin. And this fibrin will form a net that is going to trap not only more platelets but also these red blood cells, calcium ions, and other white blood or other cells like white blood cells to form a blood clot. So this is the coagulation mechanism, while this was the platelet plug formation. So the most immediate step towards preventing blood loss is the vascular spasm, and causes for vascular spasm will be activation of pain receptors that are directly found innervated by nerve cells that are going to lead to this constriction of the blood vessels, and then you have a smooth muscle injury within these cells, smooth muscle cells that surrounds these blood vessels, and also you have the platelets releasing this chemical serotonin that is going to lead to vasoconstriction. The effects of the vascular spasm is going to be the prevention of bleeding, and these provide time for other two clothing pathways to happen. So the platelet plug formation, and then the actual clotting formation. So in the platelet plug formation, you have this smooth muscle cell, which is the endothelium, the smooth endothelium inside the blood vessels, coated with this chemical. This is known as prostacyclin. This prostacyclin acts as a platelet repellent. Once you break down this endothelium, you have collagen fibers exposed into the inside of your blood vessel. And then your platelet is going to sense this exposed collagen, is going to uh, activate, and is going to extend these pseudopods to stick to this damaged area so it won't be swept away with the circulatory system, within the circulatory system. And then the pseudopods will contract, will draw together a platelet plug, and then the platelet will start degranulating, releasing substances like serotonin, ADP, thromboxane A2, which are going to act as vasoconstrictors, and they're going to attract more platelets, and then you have this positive feedback cycle that will be active until the break in the small vessel is sealed. In the clotting mechanism, which is the last and most effective defense against bleeding, you will convert fibrinogen, which is fragments of protein. They're going to clump together, form a thread of fibrin, and they will form a framework. These fibrin threads are going to form a net or a framework, and then you have the activation of clotting factors or procoagulants that are produced by the liver but are present in the plasma, and then you activate a cascade. You have an extrinsic pathway and intrinsic pathway that ultimately will merge, and then they will form this important mechanism that will form the blood clot in a stronger way to prevent bleeding. So this is the figure showing you the pathways for coagulation. So you will have the extrinsic and intrinsic mechanism that will merge in this area, and all of these are going to activate this fibrinogen to form fibrin, and then these fibrin threads are going to form this net that are going to trap more platelets. They're going to trap red blood cells, white blood cells, calcium ions, and then you have the formation of a strong blood clot. So the intrinsic mechanism of coagulation is activated by the platelets, while the extrinsic mechanism by an injury. And you have all these proteins, these procoagulants, that are formed by the liver, but they are not active unless you have an injury or your platelets activate this coagulation cascade that, as I told you, ultimately they will merge at this step, and then you have to activate this fibrinogen to form fibrin threads, and then the fibrin threads will form this net that will help to form this blood clot. So the extrinsic pathway again is initiated by damaged tissue, and then the intrinsic pathway is initiated by the platelets, and calcium is going to be important for both pathways, the extrinsic pathway and the intrinsic pathway. And you have all these proteins, these procoagulants, that are going to be activated in this sequence that ultimately will lead to the formation of fibrin threads. Okay, so this is the end of this presentation. Before we conclude, let me tell you about specific key points that are important for you to know for part of this chapter. And this will happen in every chapter. I will tell you the key points. Sometimes can be towards the end of the chapter, sometimes can be towards the middle, or any anytime within the presentation. The important point is that you need to basically pay attention and listen to the entire presentation, because the key points can be found anywhere and everything in the presentation is important. Okay, but so then here are the key points. So if someone has an infection, like a wound infection in the foot or anywhere, the person will have elevated number of neutrophils. That's one key point. A second key point is that actually there is an enzyme from the plasma that can digest fibrin to dissolve blood clots. This enzyme is known as plasmin. You cannot have, once you have an injury and you start forming in your blood vessels and you start forming these blood clots, you cannot keep these blood clots forever. You need to dissolve them. And this enzyme that can dissolve or digest fibrin is known as plasmin. So plasmin dissolves fibrin to dissolve the blood clots. Okay, so that's the second key point. The third key point is that the cells that can function as phagocytes or eat other cells will be the neutrophils, monocytes, and eosinophils. So neutrophils, monocytes, and eosinophils act as phagocytes, or they function as phagocytes, cells that can eat other cells or debris. Fourth key point is that the ion that it is found at the center of the heme molecule will be iron. Fifth key point, the origin or the cause of sickle cell anemia is a mutation or an abnormal gene in adult hemoglobin. So a mutation or abnormal gene in adult hemoglobin. The next key point is, what is the function of the erythrocytes? Carry CO2 from the cells to the lungs and carry oxygen from the lungs to the cells. The next key point is, after the first pregnancy with an Rh positive fetus, if a female gets a second pregnancy with Rh positive fetus, if it's of course an Rh negative female, the baby will develop hemolysis of their its red blood cells. This is known as hemolytic disease of the newborn. The next key point is that you will have hemolytic anemia in the newborn when the mother has any blood group ABO, as long as she's Rh negative type and the baby is Rh positive type. The next key point is that a tissue plasminogen activator, or TPA, it's a chemical that can activate plasminogen, transforming it into plasmin. And this chemical, TPA, is useful for dissolving blood clots in the coronary arteries because plasmin can dissolve the fibrin in the blood clot. So again, a TPA, or a tissue plasminogen activator, is a chemical that can activate plasminogen, transforming it into plasmin. That's why it's called the tissue plasminogen activator. And the TPA, the tissue plasminogen activator, is a chemical useful for dissolving blood clots in the coronary arteries because plasmin can dissolve the fibrin in a blood clot. The last key point, when a person's blood agglutinate with anti-B sera but not with anti-A or anti-D sera, it means that the person's plasma has A antibodies. Again, for this last key point, when a person's blood agglutinates with anti-B sera but not with anti-A or anti-D sera, it means that the person's plasma has A antibodies. Okay, so this concludes this presentation. Feel free to email me any questions, and have a good day or have a good night. Bye-bye.